Hello, everyone. Thank you for joining us today for our ALS Webinar Wednesday series. My name is Malia Forbes, and I am a marketing coordinator for ALS North America. I'll be facilitating the webinar today. Before we begin the presentation, I have a few helpful notes. If you are having technical issues with your webinar platform, type your message or question in the chat pod at the bottom right. You can also select the hand icon to notify me of your status so I can assist you. All questions regarding the webinar material will be answered at the end of the presentation. Our presenters today are Jeremy Burns and Michelle Michalchuk. Jeremy Burns has a Bachelor of Science in Biology from the University of Manitoba. He has been with the ALS Limnology Department in Winnipeg for almost 13 years and the primary analyst of algae and zooplankton in the lab for the last 10 years. Michelle Michalchuk has a Bachelor of Science in Biochemistry from the University of Manitoba. She has over 20 years of laboratory experience and has been involved in sample preparation and analysis for most of the tests offered by the ALS Winnipeg Organics section. Michelle is also actively involved with Manitoba Environmental Industries Association. Jeremy and Michelle, thank you for taking the time to deliver this presentation. I will now turn it over to you.
All right, thank you very much. Good morning, everyone. My name is Jeremy Burns. I am, like Malia said, I am the phytoplankton algae analyst here at ALS Environmental. I want to, the focus of my discussion today will be on cyanobacteria for my part of this presentation, otherwise known as blue-green algae. We're going to take a brief look at what cyanobacteria is, what some of the impacts are that it can have on water quality, and some of the guidelines and what some of the guidelines are for recreational water exposure. And we're going to touch on some of the reasons for testing for cyanobacteria cell counts. We will also look at what is involved with our cyanobacteria analysis that we do here, from the sampling materials we suggest to how we analyze the sample and what is reported to our clients. So what are cyanobacteria? They are photosynthetic prokaryotes. Prokaryotes meaning they lack nuclei and membrane-bound organelles, similar to bacteria. Unlike other photosynthetic bacteria, though, they possess chlorophyll A as their main photopigment. They also have accessory pigments like phycocyanin and phycoerythrin that mask the green chlorophyll A and can give the cyanobacteria a wide array of colors, such as gray, blue-green, violet, red, and dark brown. Cyanobacteria are found in many diverse habitats. They have been found in extremely cold places like lakes in Antarctica, and really hot places like hot springs and even some soils in hot deserts. They can also be found in fresh, brackish, and marine waters. Cyanobacteria are also very morphologically diverse. They can be unicellular, filamentous, or colonial. Here are a few examples of how diverse cyanobacteria can look under the microscope. These are some pictures I've taken myself. They're at 400 times magnification under my microscope. In the top left, you can see Gomphospheria, which has a very spherical colony. On the top right, you have Merismopedia, which produces very flat, almost 2D colonies, and very small cells. In the bottom left, you have Lyngbia. It has very compact, very thin cells, along with a sheath that it travels in, or that encoats it. And then on the bottom right, we have Rhabdoderma, kind of a very loose colony that's in a colonial mucilage. It's also in a bloom of a Phanozomenon. So let's look at some of the impacts cyanobacteria can have on water quality. So some species can form large harmful algal blooms in the water that can reduce water transparency, affect the overall biodiversity of the area, and may cause fish kills due to oxygen depletion. Contact with high densities of cyanobacteria cells found in blooms can cause inflammatory health effects, such as rashes, ear and eye irritation, and cause respiratory or gastrointestinal distress. Cyanobacteria blooms can also be a danger to pets and livestock. They can poison them, sometimes even killing them. Many species of cyanobacteria produce taste and odor compounds that are unpleasant, and they can be difficult to remove from the water without ozone or granulated carbon. And some species of cyanobacteria produce toxins. Some examples of these toxins are microcystin, which targets the liver, and anatoxin A, which is a neurotoxin. Michelle will be talking more about microcystin and how we test for it in the next part of the presentation. So these are three bloom-forming genera. We have Anabina in the top left, Phanazomenon in the right, and Microcystis in the bottom. We see these regularly in samples we analyze. They contain many species that are capable of all three of the impacts we just discussed. Many of the species in these groups are toxin producing. Some species of Anabina and Microcystis are known to produce microcystin, among other toxins. And some of Phanazomenon species produce cytotoxins and neurotoxins like anatoxin A. I'm going to focus now more on the recreational water guidelines proposed by the World Health Organization that involves the use of cyanobacteria cell per milliliter counts to help in a risk assessment of public sites. They put out a publication back in 1999 called Toxic Cyanobacteria in Water. It contained a series of guidelines for safe practices related to recreational water that is associated with an increase in severity and the probability of adverse effects. So it's based on probability because individual sensitivities can vary greatly. Let's look at the first level. Relates to relatively mild and or a low probability of adverse health effects. So they've given this level a guideline of 20,000 cyanobacteria cells per milliliter. This level is for protection against irritation or allergenic effects. The second level relates to a moderate probability of adverse health effects. The guidelines for this level is 100,000 cyanobacteria cells per milliliter. At this level, the probability of irritative symptoms is increased, and there is the potential for health impacts from cyanotoxins. The 100,000 cells per milliliter limit is also an important warning for the potential of rapid formation of scums or harmful algal blooms. Concentration of cyanobacteria at this density by wind or their inherent cell buoyancy can lead to rapid scum formation in a matter of hours. The third level relates to the probability of a high risk of adverse health effects. This level is mostly associated with cyanobacteria scums, so that are very visible in the water, and they can reach much greater than 10 million cells per milliliter. So why test for cyanobacteria cell counts? Well, cyanobacteria cell counts can be a proactive tool that can give warning to help protect people before toxins are produced. I'll also mention that early detection by a cell count may also provide a cost savings by helping to avoid expensive treatments that may be required when a bloom breaks out. In Health Canada's guideline for Canadian drinking water quality, in the technical document on cyanobacteria toxins, they proposed that monitoring cell counts or biovolume estimates in source waters, when integrated with toxin information, for example, microcystin analysis, can contribute to a better understanding of treatment requirements, potential optimization needs for plant operations, and removal efficiencies. So above are some links and resources for you from specifically the WHO publication that we talked about, the Health Canada guidelines for Canadian drinking water quality. For more information on the specific causes and management of harmful algal blooms caused by cyanobacteria, I would suggest looking at the first link there, the CyanoHabs page from the EPA. It's very well organized and it's pretty user-friendly. Also, many states and provinces, at least in North America here, also have their own individual guidelines for acceptable levels of cyanobacteria in drinking water and recreational waters. So I encourage you to check with your local government's water quality division for any recommendations that they might have. Okay, I'm going to move on to describing a little bit about what we offer here at ALS for cyanobacteria analysis, specifically in regards to cyanobacteria cell counting. So when you have a sampling requirement, ALS, we're able to provide you with appropriate containers upon request. For this test, we can provide 250 milliliter polyethylene bottles for sampling. The preservative that we use for this test is a concentrated iodine solution called Lugol solution. We can provide a 30 milliliter dropper bottle for preserving samples in a field. A 250 milliliter sample only really requires about 3 to 4 drops, or until the sample is the color of weak tea, similar to the bottom right picture there. One dropper bottle that we provide is usually good for around 30 to 40 samples. We don't require large amounts of water, so we don't need 2 liters of water. We're really only looking at a few milliliters under the microscope. So 250 milliliter is an appropriate amount, but we would probably say 100 milliliters would be considered the minimum amount of sample that we would want. We do encourage our clients to get the proper containers and preservative from us, and not to try and use their own sampling containers if it can be at all avoided. So for sample collection, the sampling strategies are dictated by the purpose of the study that you're performing. Clients are responsible for determining the strategy that meets their needs. We're not going to propose anything for you, so that's up to you, the clients. But we do recommend looking at the standard methods for the examination of water and wastewater from APHA for guidance. Specifically, Section 10200B outlines some various sampling procedures and general information to take into consideration when sampling. So hold times and submission to the lab. So there is no hold time once the samples are preserved. Once samples are preserved, they can be kept at room temperature, but preferably kept out of sunlight, direct sunlight, because that can degrade the preservative over time. That usually takes a while, but just to be on the safe side, if it takes longer to get to us, just keep it in a little cooler or something like that. Preservation is best performed in the field, but if that isn't possible, keep the samples cool during transport using a cooler and ice packs, and transport to the lab as soon as possible, and we'll preserve them once they get into the lab. This is an example of a chain of custody that we can provide. We can send these to you pre-filled in, so all you have to do is add your sampling IDs, dates and times, or we can send you an electronic copy to fill out. Just please make sure that it's very important to fill out the contact information along with an email and phone number so we can reach you properly. And if you have any specific needs for when you need it, if you need it sooner than later, you choose a priority level, let us know when you need it by. We do the regular turnaround time for this for cyanobacteria cell counting is 7 working days. But if you need it sooner than that, we can provide priority or emergency and sometimes same day. Due to the settling time for cyanobacteria, we usually can't do same day analysis. It depends on the sample. There is an extra cost, though, associated with faster turnaround times. We also need an identification for your sample. This is how your sample will appear on the final report. So be specific in case you have to look back on these reports months from now and need to know where the sample is from. Please provide sampling date and time. Usually use 24-hour time if possible, and sample type, which for this type of test, it's usually just water samples. If you've received a quote for the work, please include the quote number or job number. This ensures proper billing on our end. It's also important to include all the people within your organization to whom this information may be relevant. So due to our client confidentiality protocols, work in progress as well as sample results can only be discussed with those people whose name appears on the chain of custody or whose names are associated with the account. If someone else calls to inquire about a particular submission, the lab cannot and will not disclose any information to those people that aren't listed in either of these locations. So unless we receive specific permission in writing, that we're allowed to do so. Okay, so once we've received the samples in the lab, we begin with the sample preparation. There isn't too much involved with this step. We're mainly just pouring a subsample of 1 to 5 milliliters of sample into a sedimentation chamber and allowing it to settle. In most cases, this requires overnight settling for all of the cyanobacteria to drop out of the water column, but sometimes only a few hours is needed. The amount we put into the chamber does depend partly on the density of the cyanobacteria in the sample. For example, a sample that is taken directly from a bloom might only be countable when looking at a subsample of 0.1 milliliters compared to a sample taken from a drinking water sample. That is very low density. In those cases, we would subsample 5 milliliters of sample. Once the sample has settled in the chamber, we use an inverted microscope to examine the cyanobacteria. An inverted microscope differs from a traditional microscope in that it has the objectives underneath. It allows you to look up at the bottom of the chamber where the cyanobacteria is settled. So cyanobacteria cell counting. So the analysis begins with an identification of all observed cyanobacteria to genus. We use taxonomic keys and years of experience. It can take time to learn how to pick out cyanobacteria from large amounts of other algae and background debris. Individual cell counts are then made for each genus. This is usually done by making a count of the number of colonies or filaments observed, and then we multiply that by an estimate of the average cell count per colony or filament. For low density samples, we scan the whole chamber and all cyanobacteria cells observed are counted. In high density samples, the cyanobacteria cell counts are estimated using a field counting method. So this field counting method, it involves counting all of the cyanobacteria in a static field of view, and then we do multiple fields throughout the chamber. They're counted this way, and the sum of the cells that we observe is put through a formula that produces an estimate for the number of cells present in the whole chamber. After this field count, a scan for the whole chamber is made to account for any cyanobacteria that we didn't find in the field count. This analysis can be very time consuming, especially when the samples contain a mix of many different genera of cyanobacteria, and especially if they're in large quantities. Reporting results. So when you receive your final report, it will include the identification to genus of any cyanobacteria observed in the sample, along with a result of the count in cells per milliliter. A total cyanobacteria result will also be included, which is the sum of all the genus counts. When no cyanobacteria is observed in a sample, the report will show that the total cyanobacteria cell count for the sample is less than 1 cell per milliliter, which is our detection limit, along with a comment. We'll include a comment that no cyanobacteria was observed. Okay, so that ends the cyanobacteria cell counting part, and I'm going to pass it over to Michelle for the next part.
Thanks, Jeremy. Good morning, everyone. I am Michelle Michalchuk. I'm one of the senior analysts in ALS Winnipeg's Organic Chemistry Department, and I'm going to be talking to you about microcystin this morning. We're going to discuss what microcystins are and where they come from, microcystin toxicity. We'll touch on some North American and international regulations and recommendations regarding safe levels for microcystin exposure. And finally, I'll discuss how to submit microcystin samples and how we analyze them in the lab. To begin with, the basics. Microcystins are cyanotoxins that are released by blue-green algae or cyanobacteria. Microcystins can be toxic to plants, animals, and humans, and ALS Winnipeg performs microcystin analysis using enzyme-linked immunosorbent assay, or ELISA. Where do we find microcystins? Well, on screen you can see a nice body of water with a fuzzy green algal bloom in it. Microcystins are released by several genera of cyanobacteria, so they can be found where algal blooms occur. And surface waters used for drinking, irrigation, and recreation may be impacted by microcystins. There are approximately 80 to 90 different variants of microcystins. On screen, I have an example of the structure of a particular microcystin. Microcystin LR is considered the most toxic and most common variant of microcystin. As you can see from the illustration, microcystins have a cyclic peptide structure and they contain 7 amino acids. Microcystins are classified as cyanotoxins because they are produced by cyanobacteria, and hepatotoxins because they target the liver. Microcystins are quite stable and resistant to biological and chemical breakdown, meaning that they can sometimes reach quite high concentrations in algal blooms. Also, it's worthwhile noting that microcystin is not killed by boiling. In animals and humans, microcystins can cause damage to liver cells, leading to hemorrhaging, tissue damage, liver failure, and death. Symptoms of microcystin toxicity can include abdominal pain, vomiting, and diarrhea. Studies suggest that adults are more susceptible to the effects of microcystins toxins than children. However, children generally consume more water per kilogram of body weight, so they have a little bit more exposure than adults typically do. Exposure to microcystins primarily occurs through consumption of contaminated water used for drinking or recreational activities such as swimming. Exposure can also occur through consumption of foods such as crops, fish, and shellfish that have been impacted by contaminated water. So crops that have been irrigated with water that's contaminated with microcystin, or fish or shellfish that have been living in a microcystin contaminated environment. There are concerns that global warming and increased pollution can result in increased algal blooms, which can also lead to the increased presence of microcystins. In Canada, microcystin and algal bloom problems are historically a seasonal issue that we see through spring, summer, and fall. But as temperatures become warmer, the time frame that we see algal blooms and microcystins is getting longer, meaning that there's more opportunities for exposure. Most provinces in Canada have encountered the presence of cyanobacteria and or microcystins in their drinking water supply during the past decade. Now, I do want to point out this isn't necessarily detecting microcystins or cyanobacteria at harmful levels, but nonetheless, it has been detected. As of 2017, Prince Edward Island and Yukon, Nunavut, and Northwest Territories are the only provinces and territories within Canada that have not encountered cyanobacteria or microcystins in their drinking water supply. Prince Edward Island only uses groundwater for their drinking water supply, so that's why they aren't impacted. And I didn't find specific information about Nunavut, Yukon, and Northwest Territories, but I believe that it's their cold climate that would prevent them from seeing the impacts of cyanobacteria and microcystin in their drinking water supplies. So internationally and locally, there are a number of different regulations and recommendations regarding safe levels for microcystin toxins in water. First, I'm going to discuss some regulations related to drinking water. The World Health Organization has established a provisional guideline of 0.01 milligrams per liter for microcystin LR in drinking water. Canadian drinking water guidelines indicate a maximum acceptable concentration of 0.015 milligrams per liter for total microcystins in drinking water. The province of Ontario also specifies drinking water guidelines for microcystin in their provincial standards. They allow 0.0015 milligrams per liter microcystin LR in drinking water. In the States, the US EPA has established short-term drinking water health advisories indicating 0.0016 milligrams per liter microcystins as safe for adults and children over 6 years of age. Now, a little bit about recreational water supplies and their regulations. The US EPA recommends cyanotoxins microcystins not exceed 0.008 milligrams per liter in water used for swimming. Canadian guidelines indicate a maximum acceptable concentration of 0.02 milligrams per liter for total microcystins in recreational water. A couple things that I wanted to point out about the regulations and recommendations are that although there are some differences in different jurisdictions, the figures are ballpark the same for both drinking water recommendations and recreational water recommendations internationally and within North America. I also wanted to point out that different jurisdictions do use different nomenclature sometimes. For example, in the list of recommendations that I've provided, we see cyanotoxins microcystins, total microcystins, and microcystins LR. In Manitoba, which is where Jeremy and I live, our provincial government conducts an extensive annual beach monitoring program, which includes testing for algal blooms, cyanobacteria, and microcystins. And the results of these testing programs are available online for people to view when they're planning on visiting a beach, and also signs about water safety can be posted in swimming areas if there's an issue. Our province follows the Canadian guidelines, and Manitoba's recreational water quality guidelines indicate 0.02 milligrams per liter as the safe limit for microcystins in water used for recreational activities. On screen, I have a sign that's posted by the province of Manitoba when toxic levels of algae have been detected, and once a sign is posted, it remains in place until levels are back to a safe level. So now that you're possibly interested in submitting a microcystin sample, what do you need to know about what happens in the lab and how to submit samples? Well, Jeremy provided some really helpful information about our chain of custody forms, and definitely that kind of information is all applicable for microcystin testing as well. And then some specifics about microcystin. Microsamples should not be collected in plastic containers. Microcystin LR, in particular in aqueous solution, can stick to plastics. Samples should be submitted in amber glass vials to protect your sample from contact with plastic and exposure to sunlight. Samples for microcystin analysis should be kept cold. We recommend refrigerated conditions, so less than 6 degrees Celsius, but not frozen, or in a cooler with an ice pack. Sample holding time is 14 days from sample collection to analysis. Our standard turnaround time for microcystin analysis is 10 days. However, priority turnaround time of 5 days and emergency turnaround time of 1 day is available for a surcharge. And 40 milliliter amber glass vials and chain of custody forms are available from the lab. At the end of the presentation, I'll be posting contact information for ALS Winnipeg, but information for contacting all of the labs is available on the ALS website. And if you have an ALS location in your area, you can contact your local ALS lab and they can provide the information you need or put you in touch with the correct people in Winnipeg for questions about microcystin or cyanobacteria testing. So what happens in the lab? An overall summary is that ALS Winnipeg is accredited by CALA, the Canadian Association for Laboratory Accreditation, for analysis of microcystin in water. ALS Winnipeg analyzes microcystin in water by ELISA, and ELISA is an internationally recognized analytical method for testing for microcystin. ELISA stands for Enzyme-Linked Immunosorbent Assay, and this is a plate-based assay technique which uses highly specific antibody-antigen interaction to detect and quantify soluble substances such as peptides, proteins, antibodies, and hormones. And as we noted earlier, microcystins are cyclic peptides, so this makes them a very suitable candidate for testing by ELISA. What happens in the laboratory testing is microcystin toxin in the sample competes with enzyme-labeled microcystin for a limited number of antibody binding sites on the inside surface of test wells. After a wash step, the outcome of the competition is visualized with a color development step. As with all competitive immunoassays, sample concentration is inversely proportional to color development. So dark color indicates a low microcystin concentration in the sample, while light color indicates a high microcystin concentration in the sample. Microcystin concentration is determined by spectrophotometric measurement using a microtiter plate reader. On screen, I have a picture of the microtiter plate readers that we use for microcystin sampling or microcystin analysis. And as you can see, there's a number of small test wells laid out in strips that are about to go into the plate reader for spectrophotometric analysis. ELISA testing for microcystin is very sensitive to temperature, light, and reaction time. The ELISA kits that we use for microcystin testing must be stored under controlled temperatures when not in use, and reagents must be allowed to reach ambient temperature before use. In fact, we use an IR thermometer gun to take temperature readings of the reagents before we begin testing to ensure that they're in the correct temperature range. Some of the reagents used for microcystin testing cannot be exposed to sunlight, and some steps of the assay must be allowed to incubate in the dark. And all assay steps must be very carefully timed to ensure the test progresses correctly. All tests at ALS Winnipeg use quality control tools with every batch of samples to ensure that we're reporting accurate results. For microcystin testing, the QA and QC tools that are included are blanks, sample duplicates, matrix spikes, laboratory control samples, calibration, and continuing calibration verification standards. These QC tools allow us to ensure that the microcystin test results we provide are accurate, and they allow us to monitor our testing process from sample preparation through to final instrument analysis to ensure everything is performing correctly. Finally, when you submit a sample for microcystin, what type of results do you get? The ELISA microcystin test that ALS Winnipeg offers doesn't distinguish between different variants of microcystins. We provide results reported as microcystin, and our detection limit is 0.2 micrograms per liter. For comparison to the water quality guidelines that I mentioned earlier, the detection limit that we use corresponds to 0.0002 milligrams per liter. So you can see the results we're reporting and our detection limit goes well below the regulatory limits. Finally, I'd like to mention that a number of the resources that Jeremy included links to in his presentation also have great information about microcystin, so feel free to check those out whether you're interested in microcystin or cyanobacteria testing. And on screen, I have contact information for our biology manager, our laboratory manager, and our chemistry manager. At ALS in Winnipeg, microcystin toxin testing falls within the chemistry department, and cyanobacteria testing falls within the biology department. Thank you, everyone, for your interest in our presentation this morning. That concludes the presentation portion of the webinar, and I think we're ready for questions if anyone has any.
Thank you, Jeremy and Michelle. We open up the forum for questions now. You can type out your question in the chat section at the bottom of your screen, and I will give everyone a few moments to get their question entered. While you are doing that, brief notes, this webinar presentation has been recorded. All participants of the webinar will receive a follow-up email once the recording is available for view. All our webinars on our ALS Global website and our ALS YouTube channel. I encourage you to find a wide range of video resources and vast webinar Wednesday presentations available there. Give everyone just a few more moments to type out a question. Oh, I don't think we have any. Jeremy and Michelle, you did a great job. Oh, one question came through. What are the typical costs for kind of analysis?
I'm sorry, I don't have pricing available information with me. I think that's something that Jeremy and I both neglected to bring with us.
Yeah, it changes every year, so I'm not 100% sure. The easiest way to find out cost information is to contact either Leanne from our biology or Wa from the chemistry. They have all the up-to-date pricing lists for all the tests. It's best to go to them or to our client services department. They'll have all of the rundown for all that.
Okay, we have another question. How do you control charting for your ELISA testing? What parameter do you control chart?
For control charting, I believe we control chart our blank results. Excuse me, our blank results, our LCS results, matrix spike results, and duplicate results. Sort of any bench for the organics department anyway, any bench QC tool is normally what is control charted.
Okay, thank you everyone. Again, be sure to look out for the recording of this webinar, and we will see you next time. Enjoy the rest of your week.